Good afternoon, everyone. Welcome to the Wednesday afternoon lecture on a Tuesday. And thank you all for being here, those of you in Missouri, and hello to all those who are watching by video, because I think we're in for a real treat here. This is the annual lecture named for Rolla E. Dyer, who was the NIH director from 1942 until 1950. That means he presided over the time when NIH became plural. He started when we were the National Institute, singular, of Health, and by 1950 we were plural because of the introduction of things like the Heart Institute and the Dental Institute and the Mental Health Institute. He was also an expert in infectious disease, so we always try to choose a Dyer lecturer who is in that space of microbiology, and we certainly have a wonderful example of that today. Claire Fraser, who I've known for many years, is a distinguished investigator in the area of the microbiome, and her topic today is functional dynamics of the gut microbiome in health and disease. She's currently professor of medicine, microbiology, and immunology, and director of the Institute for Genome Sciences at the University of Maryland School of Medicine. She did her undergraduate work at RPI, got her PhD in pharmacology at SUNY Buffalo, was then on the faculty at Roswell Park until coming, guess where, to NIH, where she was from 1985 to 1992 in NINDS and then in NIAAA, rising to become a section chief of molecular neurobiology. And then in 92, she moved out there, not far from here, to be vice president, and then by 1998, president and director of TIGER, the Institute for Genomic Research, where many exciting things happened related to uncovering genomic aspects of microbes. And since 2007, she has been in this position I just named a moment ago at the University of Maryland in Baltimore. She's an elected member of the Institute of Medicine. She has been inducted as a member of the Maryland Women's Hall of Fame. She's a Burroughs Wellcome Fund Visiting Scientist Professorship awardee. And it's a great pleasure to have her here this afternoon to talk to us about something that is happening and I think causing a great deal of interest across many different fields, the way in which the gut microbiome plays a role in health and disease. And there's hardly anybody better to come and talk about that than Dr. Fraser. So please join me in welcoming Dr. Fraser.
Thank you very much, Dr. Collins. I am absolutely delighted to be here, and that was a very generous introduction. And as was mentioned in my introduction, I have been involved in microbial genomics since 1995. This is actually the 20-year anniversary of the completion of the Haemophilus influenzae genome. And when I think back to those days, in many ways it seems like yesterday, and in other ways it seems like a lifetime ago. But what I'm going to talk with you about today is work from my lab that has really represented a natural evolution of these studies from focusing on single microbes that we can grow in culture to now studying complex communities in various environments. And the environment that's of most interest to me is the human GI tract. So let me begin. Something happened that helped. This does not bode well if I haven't made it past the first slide. Well, while we're getting technical help here, let me continue and just ask you to think for a minute. When do you think the notion that we coexist with a complex community of microbes first appeared? I'm not going to ask for a show of hands. I'm not going to ask for people to throw out timelines, but you may be surprised to know. Okay, is this? You may be surprised to know. I'm going to assume that. Yes, great. Thank you, thank you, thank you. I won't touch. Here, let's get rid of that. I don't want to touch that. Okay. So here we are, and here's the answer to that question that I just posed. It was actually Antonin von Leeuwenhoek that first described the microbes that live in association with humans. one of the first samples that he examined after his construction of the light microscope was a sample of material that he scraped off the surface of one of his teeth and suspended in solution. And he wrote about what he saw in a letter to the Royal Society of London. And he was in awe of the large number of what he called small animalcules that he saw in this sample. And in fact, that there were so many of them in the water that the water seemed to be alive. So that was a long time ago, and I think it just goes to point out that very often in science it can be difficult to have an original idea. But for the better part of this 350 plus years since von Leeuwenhoek's first description of the oral microbiome, at least morphological description, I think we were very much in the dark about what this community of microbes that lives in association with humans does. It was all thought to be rather mysterious, and this in large part reflects the fact that we really didn't have the tools to study these communities at a molecular level like we do today. For a long time, this was described as the normal flora, which we don't use that term anymore, flora referring to plants. But the thinking was that our microbiome was mostly inert, it was relatively stable, it wasn't relevant to disease, and when we were thinking about microbes that were important for human health, the only thing we really needed to focus on were the pathogens. All that has changed in the past 10 years or so. And I think we owe a great debt of gratitude to the vision of Dr. Collins when he was then still director of the Genome Institute in assembling a group of people to begin to put together a plan and construct a vision for what a project might look like to study the human microbiome. And this would be a project that would make use of all of the capabilities, all of the firepower, all of the human capital that had been assembled to complete the Human Genome Project and then go on to begin to look at additional genomes, the functional units of the human genome and the ENCODE project. And we knew that these same approaches that my colleagues and I had been using could aptly be applied to the study of microbial species on Earth. The estimate is that there may be as many as 10 million of those species. Very few of them, in fact, are pathogens. Most are beneficial. And so I remember being present at what I think was the very first human microbiome planning meeting. It was at a hotel in Rockville on the eve of Valentine's Day. Many people who were there were bemoaning the fact that they weren't home with their loved ones. But it was a collection of really interesting people with expertise in microbiology, genomics, and ecology. And it really began to construct a framework for what would become the Human Microbiome Project that was officially launched in 2008. And this project was designed to use culture-independent methods to characterize the microbial communities living within us, both on all the surfaces and cavities of the human body. And you'll note on this slide that I have culture-independent highlighted in red. That was a very important aspect of this project because we know, and the field of microbiology has known for a very long time, that if you go out into the natural world and try and take complex communities from their habitat and grow them up in the lab, you fail miserably. At best, you can expect to culture 10%, sometimes not even that large a representation of what's there. It's likely because these are interdependent communities of organisms, and we don't really know how to capture the conditions that are necessary for growth of all of them. But the tools and the technology, the approaches in 2008 had absolutely matured to the point where we could take a starting sample, whatever it be, whether it be a human sample, soil, ocean water, extract the nucleic acids, and use the tools of genomics to begin to get tremendous insights about who was living in that community. And so that's what we began to do collectively, and I should mention that at the same time there was another project going on in Europe, the MetaHit project, that was focused just on the human gut, that also has added tremendously to our body of knowledge. But the Human Microbiome Project focused on 5 major body sites, and I'll get to those in just a second. This was a fantastic consortium that functioned well for 5+ years. There was a great focus on understanding the communities in a significant number of healthy subjects. There were also demonstration projects that I was involved in that were focused on trying to demonstrate whether or not there were links between altered microbial communities and particular diseases. There was a data analysis and coordination center led by Owen White. As was the case with the Human Genome Project, a focus on technology development, computational tools, and a number of ELSI projects as well. I think by all measures we can say that this was a resounding success, and I was delighted to be part of the project. And so I think we now know, we now have a very different view of the human microbiome than we did prior to all of this effort. And I would say that today we really understand that this is a co-evolved ecosystem that results in a mutual benefit for both us as the host and benefit of these commensals that share our space. I tend to take a GI-centric view of the human microbiome, but there's also been focus on the oral cavity, the respiratory tract, the skin, the vagina, and all of these are equally important. But in the GI tract, we know now that our microbial partners contribute to the digestion of our food, provide energy for our metabolism, make a number of essential compounds such as vitamins, short-chain fatty acids, can also make a number of toxic compounds. And I thought it was interesting in the news yesterday, the WHO recommendation to avoid processed meats because they have been deemed to be carcinogenic. Well, I would argue that a large reason that they're carcinogenic is because of the action of the gut microbes in breaking down the components that you find in processed meat. A really important role for the gut microbiome is in the development and maintenance of the immune system, and we'll touch upon this in just a bit. There's also a growing interest in better understanding the gut-brain axis because in a number of animal studies, the gut microbiome has been shown to influence both neurodevelopment and behavior. And I think in all human environments, a shared function of the microbiome is to act as a first line of defense against pathogens. And in the spirit of Dr. Dyer, this is the topic here that I'm going to focus on today and talk to you about some of the work that's been going on in my lab. Just a couple of other thoughts about the microbiome before we move on. It's believed that about only 10% of the cells in the human body are human. The rest are microbial cells, and about 99% of that microbial mass is located in the GI tract. And if you make some assumptions about genome density and genome size in bacteria and other microorganisms, we can do the calculations and quickly realize that the microbiome likely contributes more than 100 times the number of genes to our biology as there are genes in the human genome. And what's interesting is that we know that a number of the genes that are contributed by our microbes encode activities that are not present in the human genome. So if anything, the Microbiome Project has forced me, somebody who's made a career as a genomics person, taking a somewhat reductionist view of biology, to rethink all of that and consider human health and disease from a much more holistic perspective that includes not only us, but our microbial partners. And just a couple more slides to set the stage. All of these studies have benefited tremendously from multiple omics approaches: metagenomics, metatranscriptomics, proteomics, metabolomics. And interestingly, we are now going back into culture. I made the point that we started this project realizing that we couldn't use culture as a first step, but there's an interest in going back into culture to try and better understand the properties of some of the interesting bacteria that have come to light as a result of these efforts. One of the most important approaches that has been used almost universally, and I just bring this up because I'm going to make reference to this kind of data throughout the talk, is the use of the 16S ribosomal RNA gene as a marker. I like to think of this as allowing us to take a molecular census and count how many of what different types of organisms are there. The properties of this gene that allow this to happen, you can see the secondary structure here. It's a gene of about 1,500 base pairs that contains a number of variable regions interspersed with highly conserved regions, the conserved regions being the peaks here. We can design PCR primers against the conserved regions and amplify across one or more of these variable regions. And with sequence analysis, then what that does is essentially provide us a barcode that allows us to distinguish with a very high degree of resolution the different types of organisms that are present in a sample. So we start with DNA, we PCR amplify the 16S gene, we get the sequence, and then search that against a number of databases that are growing all the time to do phylotype classification and compare community composition across samples. So what we have come to really understand is that our body represents a microbial ecosystem. It serves as a scaffold for a wide range of different microorganisms, primarily bacteria, but also archaea, viruses, and microeukaryotes. And data from the first series of studies funded by the HumanMicrobiome Project reported in great detail the differences in the composition of these communities that are associated with different body sites. And these almost become predictive of a body site. I would venture to guess that you could show somebody who is familiar with this data, overall composition, and they could probably tell you with a high degree of certainty what sample had been studied. And I think also when we consider thinking about all of these results, we really need to remember that what we are studying is an ecosystem. And a number of the principles of microbial ecology have been shown to apply, and we're going to talk a little bit about the idea of stability and resilience as a property of these microbial communities. Other data from the HMP demonstrated that while there can be a tremendous amount of variability in who's there, and this is data shown at the phylum level in different samples from different environments in the human body, and these represent just a series of stacked bar graphs from hundreds of samples. When you look functionally at the predicted metabolic pathways that are encoded in metagenome data, you see that a lot of this variability tends to disappear, suggesting that while these communities may differ in terms of function, in terms of composition, they are much more similar in terms of their function, and that makes sense if we believe that these functions are critically important for human health. Another idea that has come out of studies in a number of different human environments, but again, I'm still focusing on the gut, is the idea that this tremendous extent of inter-individual variability can be simplified somewhat. And it was first described in 2011, the idea of community types or enterotypes as a framework that can be useful in studying differences in these communities. And what's captured in this enterotype concept is the idea that if you were to sample the gut microbiota in each of us, you would find that each of us was dominated by the presence of a single bacterial genus. The original report suggested that there may be 3 enterotypes. I think the number is probably greater than that, but it's not infinite. And that, I think that can be helpful. What we know is that these enterotypes tend to be stable over time, and there are lots of factors that seem to be at play in determining what your enterotype might be. These include host genotype, diet, intestinal transit time is one of the most recent reports that I've seen, and there are likely other factors that contribute that we haven't figured out yet. Over the course of a lifetime, we know that in the very young, these communities that are developing, they are fairly low diversity, very, very dynamic. You reach adulthood, these communities mature, they tend to be more diverse, fairly stable, and then with old age, these communities tend to lose diversity again. We also know that there are a number of factors. Diet, I've mentioned before. Antibiotic treatment is another big one that can influence these communities. Disease, there are many, many links between changes in the microbiota and disease. And I think that this gives us a framework for thinking about how we can interpret data in the context of a lifespan. So now with that as background, let me get into the meat of my talk, where we're going to focus now on pathogens and their interaction with the gut microbiota. This is a fabulous figure that I took from a review article published a couple of years ago. And what it is trying to depict here are what we think some of the differences might be between a healthy or an intact microbial community in the gut and a disrupted community. Let's begin on the left, looking at a healthy microbial community. We know that the microbiota sits in fairly close association with the epithelial cells that line the GI tract. Those cells are covered with a thick layer of mucus. Some of these commensal microbes are present in association with the mucus. Some are in the lumen. And there is a balance that exists between our commensal microbes and the gut-associated immune system that lies right below this epithelial cell layer. We know that there is crosstalk in both directions, that the microbes can have an impact on the adaptive immune response. The host can also stratify these microbes in the lumen, particularly through the secretion of IgA. And when this system is in balance, even though there may be exposure to exogenous pathogens, they are of little effect. Contrast that with what we see in the right-hand side here. A disrupted microbial community could be disrupted through the administration of antibiotics. There's been a lot of focus on the effects of antibiotics on these communities, and I think it's probably analogous to dropping a nuclear bomb on this ecosystem. You can end up with a depleted microbiota. Some of these pathogens that may be present or passing through can overgrow. There can be epithelial cell damage, which now leaves open the lumen of the GI tract to the gut-associated immune system. You can get systemic dissemination of both pathogens and commensals, and this can often drive a very robust inflammatory response. So I have been very interested in trying to figure out exactly what happens in going from a healthy community to a disrupted community, particularly interested in asking whether or not how easy is it to go in the other direction, to go from a disrupted community back to an intact community. The other thing that I should mention is that when you end up with a disrupted community, it's not just the host and the microbiota that can play a role in the outcome of infectious disease. A number of pathogens, in this case Salmonella and enteric pathogen is noted here, can use their own strategies to further disrupt this system. So there's a lot going on. And I've been very fortunate to have an opportunity to be involved in a number of collaborative studies with Marcelo Stein and colleagues at the Center for Vaccine Development at Maryland to begin to look at this interplay between the gut microbiota and enteric pathogens. And I'm going to tell you a couple of stories today. These are still preliminary studies, and we have some intriguing results, and they've raised far more questions than we have answers for. But it's beginning to give us some insights, I think, and to suggest that this interaction between pathogen and microbiome and host doesn't necessarily operate the same way in every context. This is work now looking at Shigella infection that was done by a former graduate student of mine, Anna Seacatz, and she's now a postdoc in Vince Young's lab in Michigan. So we know with Shigella, there are 4 serotypes of Shigella that can cause disease. Shigella attaches to colonic epithelial cells, enters the cells, and there is a well-described cascade of effects where Shigella can ultimately get into the bloodstream. The cause, Shigella, is endemic worldwide. It's responsible for about 120 million cases of dysentery each year. There are a couple of live attenuated and inactivated vaccines against Shigella that are in various phases of clinical trials. But one of the things that has really been a problem, not only with development of Shigella vaccines, but other vaccines, is the fact that vaccines, we know, show different efficacy across various populations. And oftentimes, these vaccines, when given particularly to children in developing countries, are far less effective than they are when given to children in Western society. And the reason for that is really not entirely clear. So we set about to study the interaction of Shigella infection and the gut microbiome and vaccination in a cynomolgus macaque model of shigellosis. First thing that we did was to characterize the gut microbiota in the macaques. This is the overall relative abundance here. And what we see is that the same organisms at the phylum level are present in the monkeys as what we see in humans, but a very, very different relative abundance. And this is just showing you the relative abundance at the genus level. We also then went on to look at all of the data that we generated in this study and ask whether or not we saw evidence for the presence of enterotypes in the cynomolgus macaque gut microbiota, and indeed we did. What we found evidence for 4 different community types. The relative abundance of the dominant members of these communities are shown here. They are clearly different. We also looked at the Shannon Diversity Index, which is a measure of diversity that takes into account the different numbers of organisms that are present and their relative abundance. And what we found was that there was a very high diversity community type 2, a very low diversity community type 3, and 2 other community types that were equivalent in diversity and in between those 2. So I've made these points. Okay. So this was the study design that was followed. And this was really looking at the impact of 2 live attenuated vaccines against Shigella that had been developed at the Center for Vaccine Development at Maryland, comparing these to a PBS control in this macaque model. There was a first vaccination, a second vaccination at 4 weeks, and then after another month, there was a challenge with wild-type Shigella. Ideally, these studies would be done in humans, but it isn't easy to get IRB approval to do a wild-type Shigella challenge, thus the use of the monkey model. The idea was then that there would be a clear winner between these 2 vaccines from this first set of experiments, and there was indeed a winner, and this vaccine, CBD 1256, was then further evaluated in a different set of animals under a different immunization regimen. Larger number of doses over a shorter period of time, again followed by a wild-type challenge. It seems simple enough in concept. My colleague Marcelo Stein was doing all of the immunology measurements, and we were doing all of the microbiome measurements to see whether immunization and challenge affected the gut microbiota. These are the data from the monkeys in study 1. These are the 2 vaccines here in PBS. And what we've color-coded here are the 4 different community types that I referred to before. Essentially, all of these monkeys started out with community type 1, an intermediate diversity community. Following each immunization, there was a transient shift in the community type to community type 3, which is lower diversity. After the first immunization, essentially all of the monkeys recovered. After the second immunization, that recovery was slower. Again, think about your ecology principles here, the concept of resilience. And this red triangle here shows the challenge with wild-type Shigella. And in that case, there was an emergence of community type 4 that persisted in a number of monkeys. You can see that these changes were relatively consistent across all of the animals in this study. When we then looked at clinical symptoms in these animals, this is what just appeared now. Green means normal stool. Yellow to red represents diarrhea, different severity. Essentially, all of these animals showed clinical symptoms. Even one of the vaccinated monkeys did. So there was evidence of clinical disease in essentially all of these monkeys. We saw the same thing in a study 3 where the animals were not vaccinated but were just challenged with wild-type Shigella. Contrast that to what we saw in the second study looking at one of the vaccines, here now in these 4 doses. In this case, the majority of the animals started out with a different community type, the high diversity community type 2, and this tended to be fairly resistant even following 4 doses of vaccine over a 7-day period, and more so even after challenge with wild-type Shigella. When you look at the second set of bars, looking at the clinical outcome with the exception of 1 incidence of loose stool or mild diarrhea in 1 monkey, there were no clinical symptoms seen in these monkeys at all. My colleague asked, what's going on? There must be a problem with this preparation of Shigella. It may not be fully virulent. After much additional experimentation, he convinced himself that the problem wasn't with the Shigella, that there was something going on here in these animals. We saw these differences, and we were very anxious to try and figure out what the possible explanation might be for the difference in clinical outcome. And one of the things that we looked at was the geographic origin of these monkeys that had been ordered at different periods of time for these two studies. And it's been known in cynomolgus macaques that there can be genetic diversity that maps with geographic origin, and that this may be related to differences in the MHC regions in these monkeys. The MHC haplotype in macaques has been shown to be important in disease susceptibility, particularly when studying SIV. And so we set out to look at genetic diversity in both the non-MHC and MHC loci in our macaque population. We looked at 24 short tandem repeats to determine genotypic diversity and hopefully relate that to geographic origin. And we also looked at 7 microsatellite regions spanning the MHC region. This is looking at the short tandem repeat data, and what you can see is that there was a group of monkeys shown here in green that were all from Mauritius that separated from the rest of the monkeys that were from Indochina or the Philippines. And comparing these data to data that had been published previously suggested to us that the geographic origin that had been suggested by the vendor was indeed correct based on the genotype data that we obtained. And I'm going to tell you, as you might suggest, it was these Mauritius monkeys that were essentially used in study 2. So we're starting to see that they're different, and they appear to be different in terms of their susceptibility to infection with wild-type Shigella. We also did the same analysis looking at the MHC allele repertoire, looking at both the class 1 and class 2 genes. And when we construct a phylogenetic tree based on that information, we again see that the Mauritius monkeys in green cluster together and apart from the rest of the monkeys in this study. So where we ended up with this initial project was that we clearly saw that there were differences there were also differences in immune response, and I'll show you that in a second, but there were differences in the clinical outcome that seemed to map with different genotype, with different MHC composition, and with different microbiota composition. It was the community type 2, the very high diversity community type that we found in the Mauritius monkeys, in which we saw no symptoms of clinical disease. So we looked further for correlations between the strength and type of the immune response and the microbiota. This is the figure that we ended up with, this network figure. It's very, very busy, and it's a little bit hard to know what's going on here, but there seems to be a very complex relationship between all of the genera that are present in the monkey microbiota, the stool score, which is the clinical outcome, and the antibody response. And so we are trying to dig down deeper into this to try and figure out what might be going on. But this first study suggested to us that genotype may indeed play a role in shaping microbiota composition, and that's not surprising. There have been a number of reports on that. It suggested to us that this high diversity community type might be protective against Shigella, but it also very much suggested to us that there may be a need to characterize the gut microbiome in future vaccine studies, particularly focused on enteric pathogens. So let me go on then and tell you another story about another enteric pathogen that's the subject of great interest at CVD, and this is Salmonella. Salmonella typhi is the causative agent of typhoid fever. It's a human-restricted pathogen that also has a severe impact on global public health, 27 million infections annually with about 1% mortality. There are 2 licensed vaccines currently available against Salmonella. And if you go back into the 60s and 70s, there were a number of human challenge studies that were done using wild-type Salmonella. Those fell out of favor, and they're now coming back. And they have been very, very important in assessing host response to infection and immunization, because as you can imagine, in the development of a vaccine against a human pathogen, the inability to do a challenge study, and this is something that we've seen most recently with Ebola, really is a limitation, because we don't always know if we are identifying a good protective correlate when we look at various immune parameters. A lot has been done in deciphering the complex immune response to S. typhi in humans. I'm not going to get into any of this except to say that this immune response appears to involve both humoral and cell-mediated immunity, and it involves a number of antigen-presenting cells as well as various effector and regulatory cells. So here was the first case study that we got involved in looking at the impact of the TY21A typhoid vaccine. This was a study to look at the effect of the vaccine on local immune responses and also on the gut microbiota. And we were curious to know whether perturbations, whether there were potential perturbations of the gut microbiota following vaccinations, and whether we could identify any associations between the microbiota and the immunogenicity that was elicited by this oral vaccine. This was the study design here. We either looked at a single dose or 4 doses of the TY21A vaccine and compared that to what we saw in a group of unvaccinated subjects. This did not include a challenge. This was just looking at immune response as an outcome. This is a summary of microbiome data using the Jensen-Shannon Divergence Index as a way to compare across samples. The size of this circle here represents differences. The largest circle here represents large differences, smaller circles, smaller differences. These are our vaccinees. These are the days over which they were followed. And I think what you can see is that the vaccination, either a single dose or 4 doses here, didn't create any consistent changes in the structure of the gut microbiota as assessed by the 16S RNA studies. We then went on to look at the cell-mediated immune responses, and this is just one piece of the data. There were multiple parameters that were examined, but these are the CD8 positive interferon production over time, days post-immunization in the vaccinees that received 4 doses of the TY21A vaccine. And what I hope you can see here is that there are 2 different types of responses. A late response that we saw in 2 of these subjects where the response doesn't peak until about day 42. And in the other 4 subjects, there was a multiphasic response where there was an increase in interferon production seen starting relatively early after the immunization here. When we looked for correlations then between these 2 different responses, the multiphasic response and the late response, and this is a heat map here summarizing the 16S data from the microbiota, we found that indeed we could separate the late responders from the multiphasic responders. You can see here in the late responders, much higher level of Bacteroides, but what was also very apparent was that there is a whole group of firmicutes here, members of the Clostridiales family of microbes that are more abundant in the multiphasic responders. And when we looked at various measures of phylogenetic diversity in these microbial communities, we found that with every measure that we looked at, these communities in which we saw a multiphasic response, which I should note is deemed to be a more robust immune response, was associated with higher diversity communities than what we saw in the individuals that exhibited just the late response. We also looked at correlations between the gut microbiota and the humoral response to TY21A, and this is using a cutoff of a 4-fold increase in antibody titer against a number of key antigens as a way to define responders versus non-responders. These are bar charts that show you relative abundance of different genera color-coded by phylum, and I think what's very apparent is that there is indeed a difference when you look at responders versus non-responders using antibody titers as a measure of the immune response. What we see is that in the responders, these tend to be much higher diversity communities. So I hope you see that there is a theme maybe starting to appear here that high diversity in the gut microbiota is associated with a more robust immune response, or in the case of the Shigella study, perhaps even a protective response against infection. We began to create correlation networks based on the antibody data. There were essentially no networks when we looked in the non-responders, but in the responders, we saw much more complex networks, and this is now looking at components of the microbiota and various immune responses. And so we are now in the process of really trying to delve into this in more detail. But we think we have some clues now, based on these kinds of data, about what components of the gut microbiota might be associated with a more robust TY21A response. And our challenge is going to be to identify the causal relationship. And we've had the opportunity to partner with Andy Pollard's lab at Oxford, who is carrying out a series of vaccination trials with TY21A and a newer anti-Salmonella vaccine. But what's so interesting about this is that they received approval to do a challenge in these vaccinees. So we're going to have an opportunity now to look at what happens when these human subjects are challenged with wild-type Salmonella. We've also generated metatranscriptome data, and what that has suggested to us in just our very initial review of that data is that in the subjects who do not develop typhoid disease following challenge, what we see in the gut microbiota is a very robust and sustained stress response that we see globally. In the subjects that end up with typhoid disease, that stress response seems to be completely missing. So stay tuned. I think this will turn out to be very, very interesting. So where we are, I think, is that we can be looking at maybe a well-balanced, high-diversity microbiota, comparing it to a low-diversity or dysfunctional microbiota, and what we may be seeing is that there are indeed some differences in overall vaccine responses or in susceptibility to infection with enteric pathogens. And I believe that we are really the first group to begin to start to look at some of these questions, and I will be the first to admit that these are going to be tough studies to try and take forward to figure out cause and effect, but we're willing to do that. And if you'll bear with me for a couple of additional minutes, I just want to leave you with another closing thought and provide some additional data. And what all of this has given me pause to think about is that if indeed the composition of the gut microbiota correlates with clinical outcomes when we're thinking about enteric pathogens, it certainly raises the question in my mind as to whether or not we can intentionally manipulate the microbiota to achieve a beneficial outcome, perhaps prior to immunization. How might we do that? Well, there are a number of ways, but I've also been involved in a series of studies to look at the effect of probiotics on the microbiota, and I just want, and that's certainly a viable, potentially viable approach to begin to think about deliberately manipulating the microbiota for beneficial outcomes. So I want to just run through a couple of slides with some interesting data that we've recently published on. This was a collaboration with Pat Hibbard at Mass General Hospital, and this was work done by Emily Elo-Fedrosh when she was a postdoc in my lab. This was part of a series of clinical trials to look at the effects of a single organism probiotic, Lactobacillus rhamnosus GG. If you're interested in this, you can go and buy this as Culturelle capsules. And this was the idea of these clinical trials was to look at the effect of LGG as a potential adjuvant for mucosal immunology in elderly subjects receiving the influenza vaccine. I'm just going to present data today from the open label part of this clinical trial assessing safety in 12 volunteers. It was a very simple study design. 12 volunteers were given 2 × 10⁻⁻⁻ LGG organisms, live organisms per day for a period of 28 days. There was then a 28-day washout period, and my lab characterized the microbiome in these subjects using 16S ribosomal RNA, some metagenome sequencing, and metatranscriptomic approaches. This is a summary of the 16S data. There's a lot on this slide, but suffice it to say that administration of LGG for 28 days had absolutely no effect on the composition of the gut microbiota. That was not a surprise to us. That had been reported previously, and LGG does not colonize the GI tract, so we wouldn't have expected to see anything. Where things got far more interesting was when we looked at our metatranscriptomics data set, and this was just a figure showing what we did. We made great use of the reference genomes that were generated as part of the HMP and mapped our RNA-seq reads back to these reference genomes as a means of doing taxonomic binning. And then once we had reads assigned to a particular taxa, then went on to do gene annotation. One of the things that we were not expecting but became very apparent as we were analyzing the RNA-seq data was the emergence of what appeared to be 3 different transcriptome groups based on recruiting reads to various reference genomes. I think of this as being analogous to what I described earlier in the talk about enterotypes defined by 16S, but this is now looking at different transcriptome groups based on functional properties, based on the genes that are actively being transcribed, and these are the 3 transcriptome groups that you can see here. We characterized these a couple of different ways. One was just looking at the dominant taxa. Group 1 was, again, a high diversity type dominated by multiple members of the Firmicutes phylum. Group 2 was a distinct group where the most predominant transcripts were derived from known gut methanogens, and Group 3 was dominated by transcripts from a single species of Ruminococcus. We also looked functionally at these 3 groups and found that based on mapping predicted genes to KEGG categories, that there seemed to be some significant functional differences across these 3 groups. If we then went on to look at the in vivo behavior of the probiotic LGG, and what you see here is this is a schematic diagram of the LGG genome. These are the 12 concentric circles represent data from each of our 12 subjects. The outer circles represent some metagenome data. But we mapped the reads in all of the data sets back to the LGG genome, and it was very comforting to see that we only saw evidence of LGG expression at day 28. At baseline and at the day 56 washout, there was no evidence of LGG present. If we look more closely at what's going on at day 28, we see that there was a strong correlation between transcript recovery and genome abundance from our metagenome data. There was also a very high level of concordance between LGG expression and transcriptome type. The 4 outermost circles in which we see the highest level of LGG expression were all from members of this high diversity transcriptome type that I just described a couple of slides ago. Interestingly, the highest level of expression within LGG across all subjects tended to be associated with a number of small non-coding RNAs, and we're not quite sure what that means other than there may be a lot of gene regulation going on. We asked at a very global level whether the presence of LGG had any impact on gene expression from some of the dominant members of the gut microbiota. We're showing here 2 strains of Methanobrevibacter, 16 Bacteroides, 5 Ruminococcus, 4 Faecalibacterium, and the answer was, for the most part, no, there were no differences over the time frame of this experiment, suggesting that if LGG has an effect, it's not a global effect. But when we looked in more detail at differential gene expression that was associated with LGG, something very interesting emerged. And what we saw looking at day 28 compared to baseline or day 28 compared to day 56 was a select set of genes that were apparently upregulated during the time that probiotic was present. And these were genes involved in motility and chemotaxis from the dominant butyrate producers known to be present in the gut. This is just looking at the data a different way, the log fold change and mapping these genes onto a schematic of the bacterial flagella and also mapping to bacterial chemotaxis. So what does all of this mean? Well, production of butyrate is one of the most important metabolic functions carried out by the gut microbiota. And Roseburia and Eubacterium, the 2 microbes in which we saw this upregulation of flagellar genes, are involved in carrying out this complex conversion of plant polysaccharides into the production of butyrate. And butyrate plays an important role in maintaining the epithelial cells in the GI tract. It's been described as having potent anti-inflammatory effects. So one hypothesis that we have with regard to potential benefit of LGG is that it may increase the level of flagellar gene expression, which then may serve to move these butyrate producers, shown here in yellow, into the mucus layer closer to the epithelial cell layer. And without changing butyrate production at all, we may be shifting the butyrate gradient in the gut, and it may result in a higher level of butyrate available to the epithelial cells. And this is based on going back into the literature to an interesting paper in 1984 by Stanton and Savage, suggesting that flagella in these motile bacteria is critically important in their being able to penetrate into the mucus layer. The other interesting possibility is that something else entirely may be going on, and we know that flagellin genes play a critical role in interacting with the gut-associated immune system. Flagellin is a ligand for the toll-like receptor 5. TLR5 recognizes flagellins from a wide range of bacterial species, and exposure to flagellin in a mouse model has been shown to suppress asthma through the generation of, or through the increase in the number of Treg cells. So it may be that this increase in the level of flagellin expression in the butyrate producers may also be having an effect on the host through its interaction with the immune system. So this is my final slide before the acknowledgments, and I'm not going to go into this in any detail other than to say, as someone who started out in microbial genomics focusing almost exclusively on pathogens and believing that pathogens are most important in directing the outcome following infection with a particular disease agent, I think my thinking and the field has certainly changed to where we're now considering all of this as an interaction triad that includes not only the pathogen, but the host and also the microbiome. So this is some of the most exciting work that I have ever done, and it's either good or bad. It's going to take a very long period of time to sort all of this out. So acknowledgments with regard to the studies that I described on Shigella and Salmonella. Folks in my lab, particularly Emily and Anna, deserve a great amount of credit. The cellular immunology group led by Marcello Stein at the CVD, and Andy Pollard and colleagues at the University of Oxford. And with regard to the probiotics study, this was also Emily Elo-Fadrash's work in collaboration with Patricia Hibbard's group at Mass General with funding from NCAM and the NSF. And I know I went very late. I have a few minutes left, and I am willing to take whatever questions you may have. Thank you very much.
We do indeed have time for some questions. There are microphones in the aisle, so please approach the microphone if you have a question so people watching the video can also hear. While they're thinking about that, Claire, let me ask you, when you propose this theme, which seems to pop up in several instances, that diversity of the microbiome is good for you in terms of presenting a better opportunity for an immune response to a vaccine, for instance, you have to wonder, I'm sure, whether that diversity is a surrogate for something more subtle going on, that there maybe is a minor species in there which is not necessarily attracting your attention, but which needs to be there at a certain level. And if you have less diversity, there's less of it, and then problems arise. How do you kind of take apart the hypotheses when there's such a complicated system under you?
That's a great question, and it's a critically important question. And, you know, to be quite truthful, I'm not sure how we go about disentangling all of this. I can tell you specifically with the cynomolgus macaque studies with Shigella, one of the things that we are looking to do now is to see whether or not we can transfer this presumed protective effect that we see with the high diversity community into a monkey with a low diversity community. There are a number of caveats, a number of challenges that will come from doing those studies. If we're successful, great. If we're not, we can probably come up with lots of reasons why that isn't going to work. And if we can do that, then we can, I think, start to systematically fractionate the communities and try and identify, is it something abundant, is it something less abundant? I don't know whether we can see this in other animal models, but this is where we're going to start, and we've thought about this for a long time, and I'm not sure how else we can do this. I think the most important next step is to demonstrate that this is a property of the microbiota that can be transferred.
Indeed. Over here.
So I have a question about the relation of the microbiome to the immune response. So how do we untangle the correlation that may exist between our immune system of the individual and therefore genetic makeup of individual and microbiome that might be correlated with that genetics versus the role of this microbiome in response to the vaccine? How do we associate causality and from that correlation?
Again, another question, and I think it really follows on the question that you raised. We have made a number of observations that there are genotypic differences, MHC differences in this monkey study, microbiome differences, and differences in susceptibility to infection with Shigella. It's going to be real. I have to say, it's going to be tough to figure out what comes first. I think we can perhaps get some insights from fecal transplantation studies. Look to try and this, unfortunately, when we're looking at Shigella and Salmonella, we don't have the benefit of doing a whole lot in mouse models. These are pathogens that are specific to non-human, human primates. So that limits what we can study. Perhaps we can find something even, a simpler, completely simpler system like Citrobacter infection in mice, for instance, just to try and demonstrate these principles. Is it the host genetics? Is it a component of the microbiome? Is it both? I think we're going to be beating our head against the wall for a long time trying to get to these kinds of answers with these studies. We have to find a way to try and do this in a simpler system. But what has struck me over and over and over again is the fact that it's these high diversity communities that seem to be associated with a more robust outcome. And I can't today tell you what's driving that, but I can't let it go. I'm really intrigued and think that there may be something there.
Right. Over here.
Hi, Dr. Fraser. Thank you for the interesting talk. I was wondering, do you know if in cases where the human biome is changed, how long that effect is retained? Does the human body, for example, automatically try to recorrect a changed microbiome back to a original template?
Yeah, that's a good question, and I would say in most cases the answer is yes. If we look at the studies that have been done in adults, there can be various perturbations. Diet's been looked at in a number of studies. And if you switch from an omnivorous diet to a vegetarian diet, you will see a shift in the microbiota. But once the diet reverts to its original state, the microbiota tends to revert as well. The one exception to that, I think, is with antibiotics and repeated exposure to antibiotics. In some individuals, you will see this restoration of community composition following administration of antibiotics, and in other individuals, after the passage of a period of time, the composition appears to be something different. And it has been suggested, and I would pose the question to the audience here: how many of you can remember how many times you've taken antibiotics over the course of your lifetime? I know I can't, and I suspect that's probably true for most of you. So it may be that, again, in some of us, with these communities that are less resilient, those kinds of exposures over time start to change the composition and presumably the function of the microbiota. And Marty, if you're interested in this, Marty Blazer has done some phenomenal work looking at both therapeutic levels of the impact of therapeutic levels of antibiotics as well as the impact of subtherapeutic levels of antibiotics. We're exposed very oftentimes in our food supply to chronic low levels of antibiotics that seem, at least in mouse models, to be having a long-term effect.
So in those cases where the biome did change, does the human body adapt to the change, or is it?
That's a fantastic question, and that's not going to be so easy to figure out, but you could postulate that as the microbiome changes and its properties change, that it may then somehow change our susceptibility or our ability to respond to a different perturbation. But to get answers to those kinds of questions would require a number of large long-term studies, and while it would be great to be able to do those, I'm not sure we can afford them.
Maybe we'll get there as it gets cheaper. We can probably just take one more question, and I know you've been waiting for a minute. So
Thank you so much for a great talk. My question is about mental health. Do you think microbiome influences mental health diseases such as depression or bipolar or schizophrenia?
Well, there have certainly been some very intriguing results from animal studies, again, to suggest that the answer to that question is indeed yes. And so by extrapolation, it wouldn't be implausible to think that the same things could be at play in humans. But again, with all of these things, we tend to be talking about a very complex biology, and we overlay on top of that the potential role of a very complex microbial community. I think it's important to always remind ourselves that if we really want to get at fundamental mechanisms, we're going to have to be doing that by coming at the problem from lots of different directions.
Yeah. Well, those of you interested in continuing the conversation are warmly invited to refreshments in the NIH library right around the corner. Meanwhile, let's thank our speaker one more time.